Latifa, I can see that we are starting to have few more attendees. Shall we make a start?
Yeah, please, that's what I was saying, but I was muted. Yeah, please go ahead. Yeah, please go ahead, Alex. Thank you.
Okay, great. Thank you, Latifa. So good morning, good afternoon, good evening, and welcome to the webinar series of the IHCC and our March offering. Today, we welcome Professor Philip Awadala, who is the National Scientific Director of the Canadian Partnership for Tomorrow's Health, or CanPath, and is the Executive Director of the Ontario Health Study. He is a professor at the Department of Molecular Genetics, School of Public Health at the University of Toronto, and Director of the Computational Biology at the Ontario Institute of Cancer Research, where he leads a research program studying both genetics and environmental determinants of early cancer evolution, aging, and studies fundamental processes sharing genomic variation. He's also on the steering committee of the IHCC. So with that, I would like to welcome Philip to talk to us about using the Canadian Partnership for Tomorrow's Health to identify the earliest determinants of disease evolution and aging. Thank you.
Great. Thank you very much for that introduction, and thank you for including me here today. So one of the key messages that I want to come from this presentation today, or hopefully will come from this presentation today, is to describe how we're thinking about population cohorts in the context of, as we heard in that introduction, early determinants of disease, aging, with a bit of a focus on cancer today. And I think this is the way we're going about it is something of a paradigm shift, if you like, in terms of how population cohorts can get used or be used, somewhat dependent on the nature of the population cohort naturally, and of course on the how we've consented our participants. So first, in today's presentation, I'm going to be presenting what our population cohort in Canada is, which I'm the National Scientific Director for. It's called the Canadian Partnership for Tomorrow's Health, or CanPath. CanPath is Canada's largest population health research platform. And then I'm going to talk about 3 or 4 kind of use cases, scenarios of how we've been using CanPath to understand, say, things like environmental determinants associated with health, as well as how we've been using CanPath to understand early development and evolution of cancers and or aging. So as I mentioned, CanPath is Canada's national platform for population health research in Canada. 1 in every 100 Canadians has consented to be part of CanPath. And within the age ranging that we are studying, it's a population cohort of adults. 1 in every 50 Canadians has actually consented to be part of the program. And it's been ongoing for about, I'd say, 12 or 13 years now. And one of the motivations for CanPath as a resource is it's Canada's population laboratory. And that's a phrase we've taken or stolen, if you like, from Terry Manolio, who had a seminal paper in Nature Reviews Genetics 2006, and also earlier papers describing population laboratories. But one of the things that we think about in the context of population laboratories is that they are a sampling of the population of both healthy and unhealthy individuals. And we gather information from individuals before they develop diseases, and in doing so, we can potentially capture important variables associated with the development of, say, cancers or chronic disease, or cancers could be some of the major ones like breast and prostate, which are our largest numbers in Canada, or things like asthma or COPD, or of course heart disease. And so what we try to do in CanPath is we try to be pan-disease or ecumenical across these diseases, such that we capture information that will support external research, exploring and studying and having hopefully a translational impact on a number of different diseases. This is a map of Canada, and this is a breakdown of our participating provincial cohorts all taking part in CanPath. And so you're getting a breakdown here. I'm sitting here in Toronto, where I also lead the Ontario Health Study, and you see there on the screen the number of participants that have enrolled. We've already, this picture could get updated because we have now Manitoba Tomorrow Project's participants now enrolled as well, and a few thousand have already started there. And our newest cohort is the Healthy Future Saskatchewan, which is in both pilot phase and of recruiting new participants participants. And you can see on the left of this figure here, the kind of information that we capture from our participants. Our participants consent to providing us through questionnaire data, information about their past, say, health histories. We capture social, other determinants of health like alcohol and smoking usage. Our participants, the great majority of CanPath participants, consent to linkage to administrative health records. And I'm going to emphasize that a lot in our future use case scenarios with regard to the examples that we're going to be talking about today. Participants provide biologics. For a good number of our participants, we've gotten physical measures. And this just gives a really high-level overview of some of the meta variables that we capture with our questionnaires. A lot of this is very typical, of course, of many population cohorts around the world. We are consented to do longitudinal follow-up, so we've already had a number of surveys, and this just gives you a quick snapshot. And in fact, during the pandemic, we've been very active in getting follow-up information of our participants as well, and that's why we call these things COVID questionnaire information. We didn't just capture COVID information from our participants, but it was an opportunity for us to also capture what we would have called medically related health information as well from our participants. So this is just kind of gives you an indication of the number of variables, over 1,600 variables related to disease outcomes or disease-related phenotypes. As I mentioned before, we capture physical measures from our participants. So participants across Canada were invited to come to an assessment center where they had, say, blood pressure measured and so on, BMI. For a smaller subset with partnership with our key collaborator, Sonia Nand, we've also been able to capture, say, imaging data. And Sonia's has significant past expertise in working with Indigenous participants in Canada. So you can see here that she had a partial focus, if you like, on recruiting and capturing imaging data from Indigenous participants as both part of CanPath and her ongoing studies as well. We heard in the introduction that I'm part of the IHCC program. I'm a member of the steering committee, and with Thomas Keane and Melanie Courteau, we're leading activities associated with data interoperability and making IHCC data available or visible to the external research community. So CanPath is also a member cohort of that activity. And I would be remiss if I didn't show the CanPath portal, which we host here out of the OICR. Again, this is work with Thomas Keane and Melanie Courteau. So for those of you who are part of the IHCC, this is a snapshot of this beautiful portal that Melanie has been supporting and maintaining out of the OICR, where individual researchers can explore the IHCC member cohorts, including CanPath, and I think CanPath is here on this screenshot, and people can explore some of the meta variables that have been collected across the world. Now, I want to emphasize going forward now what CanPath can enable given our consented program, which is our ability to go forward in time capturing information about our participants, either through, say, recontact of participants directly with surveys, capturing more information with regard to blood variables if we collect more blood, but also all of our participants have consented, as I mentioned before, to administrative health records. So we, through various entities across Canada, can link out and capture, say, electronic health record related outcomes, for example. So at a high level, say, for example, or an example would be, say, ICD codes. And that allows us to think scientifically, both retrospectively as well as prospectively. We can look into our population cohort, say perhaps for some of our healthy participants, and ask what has happened to them since we last saw them as part of the cohort. Who developed a disease? Who didn't develop a disease? And then explore the cohort and ask why. Some of the things that we can also look at is say things like environmental data. All of CanPath participants, our consent allows us to link out in Canada 6-digit postal codes or addresses and use that information to link to environmental databases like the CANU database, which allows us to explore, say, at 6-digit postal code resolution, potential environmental exposures and how that might be associated with health outcomes. So, for example, we could use, say, some of the clinical information that we've been capturing, maybe some of the functional omics or genomics that we've also been capturing, say, gene expression data, genotype data, and then link to, say, CANU to capture some of those environmental exposures and ask, what are some of the most important determinants with respect to gene and environment on health outcomes. So just a quick example, we started a project within, say, Quebec, where we were able to look at different, say, urban centers with varying levels of density and varying levels of, say, environmental or pollution exposures, Montreal, Quebec, and in the north of Quebec, Saguenay. And Quebec is a great kind of exemplar of how founding populations are useful for, say, controlling or looking at the impact of founding populations, and genetics, where we know a great deal about the history of how genetic movement has helped populate Quebec, and use that information to understand those types of questions, like how much gene versus environment has impacted the health of, say, Quebecers. And so this is just a quick snapshot where we say use genetic ancestry information of, say, Quebec, and using information that we have there with regard to that founding population. We've done gene expression or RNA sequencing profiling of a good number of participants from those 3 environmental varying regions. And then also look at, say, which environmental variables like PM 2.5s, particulate matter, air, ozone, green population density, walkability indices, all the kinds of information that were captured in CANOE, and ask what explains, say, the gene expression variability, for example, that we saw, and how that relates to chronic disease. Is it genes or environment? And just really quickly, this is a publication. I'm not going to have time to get into this too much today. We published in Nature Communications, I think a number of years ago now, that gene expression profiles largely reflect the region individuals live in rather than their genetic genotype or their genotypic ancestries. And the gene expression signatures that we were seeing in those 3 different urban centers were largely driven by the types of environmental exposures people were exposed to, for lack of a better term. So particulate matter 2.5 came up, NO2, lots of those types of exposures were the driving impact on some of the gene expression variability we saw in those regions, and largely linked to, say, things like cardiovascular disease. And just to round this off, we took that information, went across Canada and asked, for example, with regard to cardiometabolic traits, what were the most important variables associated with, say, some of the variation we saw at the protein level with regard to the health of Canadians. And I just highlighting a red arrow here is that the third most, I think the actual second most important variable with respect to, say, some cardiometabolic traits after age was particulate matter 2.5. You can see here a panel of, say, cytokine profiles that we've been also exploring here. They also explain some of the variation in the with regard to cardiometabolic traits in the population. But it's quite remarkable to us that PM 2.5 ranked number 2 after age, of course. Okay, so that's a large kind of overview of what CanPath can enable in the context of what we've captured with regard to samples from across Canada, from over 300,000 participants, our ability to link to, say, environmental measures, the kinds of stuff we're doing with regard to genomics and, say, transcriptomics. Now, what I really wanted to focus in on now is how we can use some of the linkages that we've been doing to help develop, say, liquid biopsies that can potentially enable us to learn what are the earliest determinants of, say, cancer evolution. And that's largely enabled because we were able to have linkage to those administrative consent resources. Just to kind of help in terms of motivation, the earliest we can detect a cancer, the better our outcomes will be with regard to treatment. And so the picture on the top right is really just speaks to 5-year survival rates. And when treatments are supported, and you can see 5-year survival rates are much higher, the earlier a cancer is captured. And the picture on the left is sort of your usual kind of description of how cancers evolve. And as I mentioned before, there's a lot, and you've probably seen quite a lot of, both in the research and the commercial space now, a lot of developments with regard to liquid biopsies. And what people mean by liquid biopsies is using, say, blood to detect a cancer without actually doing something more invasive like a tissue biopsy, say with regard to, say, breast, prostate, or many other cancers as well. And a lot of those liquid biopsies use cell-free DNA to discriminate, say, DNA captured in blood from that might be shed from a tumor somewhere in the body relative to, say, DNA that might be shed in blood normally. And so one thing that can be done, and this is what we've been using, is that the DNA methylation signatures of cell-free DNA is very tissue specific. And so we've been training models using cancer repository data, like the TCGA repository, to train models such that the DNA methylation signatures that we capture in liquid biopsies could detect where a cancer is coming from in plasma of a participant. We started this project with individuals from the Ontario Health Study, which is part of CanPath, and as was mentioned during the intro, I'm leading out of the Ontario Institute for Cancer Research, where we have 40,000 blood plasma samples. And through our linkages to administrative data, we're able to identify participants who came into the cohort healthy, say in 2009, 2010, all the way up to 2017, and then have since developed a cancer. And we would know that they developed a cancer because we link out to, say, Cancer Care Ontario registries. So we have registry data access that allows us to identify, can we see plasma samples and then interrogate those plasma samples for the potential signature of a cancer before they were actually diagnosed. And that's a take-home message I want to show here. So you can see on the bottom right, some information here in terms of follow-up time. So things would have been, are at 0 when a plasma sample came in, and then anywhere from a couple of months to maybe up to 7 years, we can see that somebody may have developed a breast cancer, a pancreatic cancer, or a prostate cancer. And I just want to emphasize this is work done by a very talented student, Nicholas Cheng, in terms of developing these profiles. And the linkage work has been driven by work by Kimberly Skeet, who's also a very talented PhD student in my group, and I'll be talking about her research in a moment. So this gives you an indication, of course, of the different stages of the different cancers that we're able to capture. So for breast, pancreatic, and prostate is where we started our project. And in breast and prostate, you can see that most of the data that we have are for stage 0 to 2. And that has a lot to do with the fact that given the screening, the mammography screening programs, say here in Ontario, we're capturing those signatures relatively early. In contrast, pancreatic cancers, despite screening, would be in the Ontario Health Study, at least, many of our pancreatic cancer cases are stage 3 and 4. And so it really speaks to the need to really detect as early as we can a signature, say with a non-invasive of pancreatic cancers before they develop to stage 3, stage 4. Again, as I mentioned before, we've developed a liquid biopsy that uses DNA methylation, as well as, and then we sequence those reads to capture those methylation markers in plasma. And this gives you just a general sense of how we're doing. You can see here that compared to controls, we are seeing really nice separation of the cancers for breast, prostate, and pancreatic. And of course, we don't have as many pancreatic cases because the incidence rates for pancreatic cases are far fewer. And in fact, actually, to do this project, we pulled all the incident cases that we had in the Ontario Health Study. And you can see that really nice in this unsupervised plot, a real separation between age match controls as well. And we try to match with things like BMI, alcohol usage, and so on. You're seeing a really nice separation of those plasma samples of pre-diagnosis cfDNA methylation signatures compared to controls. And what we can also show, and I'm not going to spend too much time on this because we're getting into the details, is that actually much of those signatures can actually separate the different kinds of cancer types. And again, we use the TCGA to do this. We've developed machine learning tools as well that now allow us to give some sort of confidence or level of confidence of how well we can detect those signatures pre-diagnosis. And one thing I want to also highlight is in many cases, we are seeing these signatures of, say, breast cancers, even just a few months before a mammography screen happened and a breast cancer went undetected. So really highlights how sensitive these liquid biopsies can be for detecting a cancer before, say, traditional approaches. Again, we can apply this to metastatic breast cancers, the overall tools. And again, what we're showing you here is that we're able to do this in the context of both prostate cancers using these same kind of machine learning tools for prediction or detection for prostate and pancreatic cancers as well. So just to summarize this work, what I really wanted to emphasize here with Nick's program is that these population cohorts, which have banked material, blood material, and keep them for extended periods of time, are extraordinarily valuable for developing these early detection tools. And you never know, particularly with population cohorts, what is the most valuable plasma sample or blood sample. So there's often a lot of talk about, well, the expense of biobanking and, why are we banking so much material? People will develop chronic diseases and people will develop cancers. And these materials are going to be critically important even before somebody has developed or even if you haven't actually sampled somebody with a particular disease, because those people will develop a disease. And they're incredibly important for developing these types of tools and validating these types of tools that are out there. So just to highlight one particular message as well, we're able to detect signatures of breast cancer up to 7 years, in fact, before a traditional diagnosis. And just a little bit more of the same here. And we can do this not just with regard to breast cancers, of course, but across a range of cancer types. Now, everything I've just talked about was actually in the context of solid tissue type cancers, but we're also doing significant work in the context of, blood malignancies as well. And so I'm now going to highlight work by Kimberly Skeed, who's a PhD student in my lab, and she also works as a national scientific coordinator for CanPath as well. So in this context, Kim's been studying how somatic mutations accrue in blood. And the reason I'm highlighting this in this particular setting is that many of the population cohorts that are part of IHCC have collected lots of blood samples, and they can be enabled, or they can enable research into how not just germline DNA variation is important with regard to the development of the disease, but also how somatic mutations in blood could be important predictors of who develops disease as well. I can't go to a conference, particularly a blood conference or even a genetics conference, where I'm not hearing about age-related clonal hematopoiesis. Age-related clonal hematopoiesis is that process by which somatic mutations in blood accrue. It's an age-dependent phenomenon, largely happening in blood stem cells and marrow. And these are the genes that are largely been seen as targeted, or as where somatic mutations largely accrue. And they offer up a fitness advantage, if you like, of those cells that have those mutations, and they clonally propagate in the population. Somatic mutations can come in two flavors. They can be point mutations, and they can also be larger variations, like structural variations, or what we could sometimes call mosaic chromosomal alterations. Today, I'm just going to largely focus on what we've been doing to capture large mutational signatures of mosaic chromosomal alterations from blood samples. And this is using tools that have now been readily used in population cohorts like UK Biobank, where people have looked at the genotyping data from the UK Biobank and using the existing genotyping arrays that have largely been used to explore germline variability in association with disease, but to call also somatic alterations that might be accruing within an individual's blood sample that may look like gains, losses, or copy number neutral loss of heterozygosity. And so this is just a real and simple cartoon of what those genomic alterations might look like in chromosomes. UK Biobank studies have largely seen that 5% of the population shows these types of chromosomal alterations within an individual's blood. And so we were interested initially in asking, why are those chromosomal alterations actually persisting? Are they persisting due to chance, or are there some other mechanism that's maintaining them in an individual's blood? So we started looking at this in CanPath, and in CanPath, we've actually been able to map, and this is just a circus plot of the map of those chromosomal alterations and where they're occurring. And we actually have in CanPath 3 different genotyping array kind of platforms that we can use to explore this. And what we were actually able to see with CanPath data is that actually closer to 14% of the population in CanPath has these chromosomal alterations accruing as you age in blood, which I thought was really quite fascinating. So again, that question as to why these things are persisting, or are they persisting? Do they appear and are actually lost with time? Are these just a snapshot, a random snapshot, and do these things actually persist, or do they actually are lost due to, within individuals' genetic drift? One thing I want to say is that the technology that we have access to, compared to, say, UK Biobank, is different. We have higher density genotyping arrays, and some of the CanPath participants, the Omni 2.5 arrays, are actually really quite critical in capturing both smaller alterations. And so we might be actually able, as a result, be able to see whether we see more of these things. So one question we did want to ask, actually, with regard to the size of these alterations is selection on these chromosomal alterations happening? And one way we can ask that is by asking if there's a relationship between the size of these chromosomal alterations and their frequency within an individual's blood. If there was no impact of selection on the size of these fragments, you might see a picture like this, where there's no relationship between the length of a chromosomal alteration and its frequency in an individual's blood, or the cell fraction. And in fact, what we don't see is that. What we do see is that there is a significant negative relationship between these chromosomal alterations and their frequency in blood. So indicating that probably negative selection is removing the larger chromosomal alterations, which makes sense. A larger genomic alteration probably impacts more genes. They're largely going to be negative in terms of fitness on the type of cell, and so are going to be removed. Across sorry about this has been cut off. We were able to map hotspots across the genome of these chromosomal alterations. So known hotspots that we were able to see were at one's aberrations on chromosome 13. These are known somatic hotspots that includes the DLU region, but we were also able to capture on, say, chromosome 2, other chromosomal hotspots. Some of these genes in this particular hotspot have been implicated already in clonal hematopoiesis, including, say, ASXL2 and indeed MT3A. And one thing I wanted to quickly highlight there as well is that the cell fractions of those mutations in those hotspots is actually higher. So it might be speaking to the fact of positive selection, maintaining those chromosomal alterations within those hotspots. So again, another key takeaway from this work, and again, something I want to emphasize in the context of population cohorts, is a lot of the classical genotyping data that gets done with population cohorts can be used to capture not just germline genotypes, but also these chromosomal alterations. I didn't have time to talk about this today, but these chromosomal alterations seem to be predictive of blood malignancies. And so here you've got a quick snapshot of the work Kim did, which is a survival analysis showing that the number of these chromosomal alterations is predictive of whether you will have a blood malignancy a lot or not. And again, something I didn't have time to talk about today is looking at the relationship between these chromosomal alterations and transcription. So the last thing I'm going to talk about today is a final kind of project, which is sort of taking a switch on thinking about disease and trying to demonstrate how we're also using population cohorts to study healthy aging. So instead of thinking about who's in the cohort getting a disease, we're also thinking about who in CanPath are our healthy agers and why, right? Who are our 80-year-olds in the cohort, our 90-year-olds in the cohort who have not developed a cancer, maybe not developed a cardiovascular disease, maybe have the blood profile of a 40-year-old, and interrogate their genomes, their transcriptomes at various levels to address why those individuals are healthy agers. Do they have a protective mechanism, or do they have just been lucky in the context of, say, types of exposures over the course of their lifetime? So this is work of a student that's now just graduated, Alyssa Bader. Now she's working for a company in California called Genentech. And she decided to take this very, I think, really interesting question at the single cell levels to interrogate just at the single cell level those individuals in our population cohort who are our healthy agers, and compare them to, say, other individuals in the population cohort. And a caveat that we would add to this type of research, and I think is an important caveat, is when you're studying the blood profiles and other profiles of, say, healthy agers, you're largely looking at a specific tissue. You're looking at blood, right? And it might be it's a very important tissue, as all tissues, but blood, does speak to all of the other tissues in your body. But you are still interrogating, say, the functional genomics and the single cell genomics of a particular tissue when you're studying these types of things. So we want, it's a caveat that we're interested in exploring is why does the blood profile of our 80 or 90-year-olds look like that of, say, a 40-year-old? So to select participants for this study, we use something called the Intermountain Risk Score, or a modification of that. And that Intermountain Risk Score is a score for predicting 5-year mortality. It includes a lot of the variables that we have captured in CanPath, like CBC information, the amount of neutrophils that are observed on a classical CBC's test, the RDWCVs, hematocrits, and so on. We made a modification because we were interested in how age might be. We were interested in our healthy ages, so we took age out as a predictor in that risk score, and used that to select participants from CanPath. So on this figure here, you're kind of getting a distribution of the proportion of participants who have that Intermountain Risk Score, or what we're calling the complete blood count risk score, and how they're distributed with age. And given this distribution, so that CR risk score will rank from 0 to 10, so 0 being low and 10 being high. And we selected from this distribution individuals that we considered aged but low risk. And the reason we were interested in that is we wanted to compare our aged low risk to our aged high risk individuals. So our people who are in that high age range, north of 70 years of age, and have blood profiles that vary between low risk and high risk, and compare them. And then in comparing them, we might be able to address this hypothesis. Are there protective mechanisms that put people into the aged low risk category? And then compare this to the age low risk to our young low risk, as well as compare them to our young high risk as well. And so we wanted to sample from the extremes of this distribution so that we can capture factors of significant impact. And so the first thing we did was we just looked at the blood cell phenotypes in those different groups and compare them. And what you can see here is that for certain comparisons like RDWCV, our aged low-risk individuals have very similar values to that of our young low-risk individuals as well. And as well, our young high-risk individuals will also have similar values to our aged high-risk values as well. So everything we're trying to do in a symmetrical kind of way. We took those individuals then, and we did single-cell RNA sequencing on about 500 of those individuals. We not only did or 400, just north of 400 individuals, sorry. We've also did performed whole genome sequencing. We also performed attack sequencing on those individuals. So, this just gives you, you can't go anywhere with seeing one of these UMAP plots of single cell RNA sequencing data now. And this allows us to interrogate at the single cell level what's going on in these healthy individuals compared to these unhealthy individuals. And this also exemplifies the biobanking that we have in CanPath, that we were able to show that we can actually do this with high fidelity with our cryopreserved samples. We looked at differential gene expression between our high risk and low risk aged individuals. And in terms of differential gene expression, we found nothing in our males. However, we found in terms of specific genes that could discriminate aged males from healthy that were different than aged low risk high risk males. But with regard to females, we did find a number of loci that discriminated high risk versus low risk aged individuals. And this is a quick snapshot of some of the genes that we saw, and we can see them at within different cell types as well. We also took another approach, similar to the approach that we used with Nick's project and the Cell-Free DNA project, where we tried to develop a transcriptional risk score to differentiate low and high risk individuals to identify potentially more genes, both in males and females, that might be associated with high risk, low risk blood profiles among our aged individuals, as well as our young, comparing healthy and unhealthy individuals. And this performed actually a lot better. And you can actually see here, these are areas using a cross-validation approach, areas under the curve for precision receiver operating curves. And both for males, females, and males, we see that for individual cell types, we can capture a number of genetic features that discriminate in a sex-specific way, healthy versus unhealthy aged individuals. And I just want to highlight a couple of things. A lot of these genes are highly sex-specific. And so what we got here on this plot here, and I don't want to spend, this is a very complicated figure, and we're not going to have time to get really into the details too much of it. On the x-axis, so we have females, and on the y-axis, we have males, and we're looking at the effect sizes of specific genes associated with low risk and high risk. And because many of these things are falling on these x and y axes, that means that they are highly sex specific. Those that do not are ones that are not as sex specific, and we see in both males and females as well. And Alyssa's highlighted here in her work some specific loci. So, for example, a number of HLA specific loci that are female specific, and a number of other loci associated with homeostasis that are also female specific. To answer the question of these two hypotheses that I laid out there before in terms of what's potentially protective versus healthy aging, this is how we broke these down. And so these genes that were captured in these transcriptional risk scores can be associated with either healthy aging in these types of plots that you see here on the left. So if something is healthy aging and is actually transcriptionally upregulated, you will see more of that in aged individuals that are high risk compared to low risk, and potentially the opposite if they are downregulated. And the opposite would be the case for protective genes, where you might see very specific loci that are not seen in the other low risk categories that are specific to a high risk or aged individual as either upregulated or downregulated. And the great majority of the genes that we capture we put into the healthy aging bin rather than the protective bin. And so that's what the pie charts on the right are describing for you here, is that the maintenance of expression levels is in the healthy upregulated group is the dominant mechanism. Now, that's not to say that the protective genes weren't important. This is just based on counts. And in fact, some of the interesting genes that are protective seem to be associated with clonal hematopoiesis, which is, I thought, fascinating, is that everything in my research program is kind of doing full circle. Everything that we're capturing around aging and healthy blood, some of that is associated with clonal hematopoiesis or ARCH, like what I described with regard to Kim's work. So just to summarize what we've captured with the healthy in the aging project, and I just again wanted to emphasize that sure, these population cohorts are important for understanding determinants of cancer risk or disease risk, but they're also critical for understanding healthy aging as well. So future directions in this space, well, as I've highlighted already, somatic mutations might be something we need to start exploring more in the context of healthy aging. For example, do our healthy agers accrue fewer somatic mutations as they age as well? So that's it with regard to the science that I wanted to highlight here. And so I'm coming now to the end of some of my presentation. So I first wanted to, of course, recognize many of the partners that have been part of CanPath over the last, say, 13 years. These include funding partners, our provincial partners, our partners that help us with regard to those linkages to administrative health records and so on. I also want to highlight that CanPath itself has a portal. I did mention the IHCC portal, which is a way for you to kind of see the variables that have been captured in an international space that we've been working with on developing at the IHCC. But CanPath itself has an access mechanism, and so much of the variables that you've seen here can be seen on the CanPath portal. This is just kind of a quick snapshot of some of the data that you can see. I mentioned earlier that the CANOE data, which is our environmental exposure data, can also be linked out through the CanPath portal as well. This is just a quick screenshot of how you can also apply for access to the CanPath. So we have a forum, we have a team and our National Coordinating Center who are very friendly and are very experienced in terms of helping support your CanPath access requests. This is the CanPath team. So of course, I don't lead this by myself. We have a national leadership team. I'm the National Scientific Director. John McLaughlin is with me at the University of Toronto, co-leading this as the Executive Director, as is Trevor Dummer, who's our National Co-Scientific Director in Vancouver. And we have, of course, all our regional principal investigators who are critical in maintaining our operations and our scientific operations across Canada. Our National Coordinating Center is at the University of Toronto as well, supporting our access to the cohort, your access to our cohort. And of course, most of the science that I just described was done by my team at the Ontario Institute of Cancer and the University of Toronto. So I just want to acknowledge specifically Nicholas, Kim, Alyssa, and of course, Vanessa and Mawusi, Vanessa Bruat and Mawusi Obezi, who have been with me for an awful long time, supporting a lot of the omics activities that we've been doing in my lab with CanPath as well. So I think that's my last slide. So I'd like to thank you for your attention today. Thank you very much.
Great, thank you so much. This was an excellent presentation. I think that we already have a number of questions. So Rongling, if we go with yours first.
Yes, thank you, Felix. Very nice presentation. Now we know the CanPath cohort much better, which is very good. And I have one question about the data access. So for the CanPath data, cohort data, the people in the or investigator in the IHCC can access based on your policy for the access of this data. If the people outside of IHCC, can they also access the data?
Absolutely. So we've already had since we've opened our doors to access both at CanPath and to the different regions, we've already had many hundreds of access requests, and they are coming from. And some of them have been coming in since before IHCC even existed, frankly. So our access policy supports external research. I would say that most of our external access is coming from Canadians, but naturally or Canadian researchers, sorry. But we're certainly, there's nothing limiting anyone internationally from having access to our data and supporting their research, whether it's with their own population cohorts or something individually with supporting their own individual research interests.
That's great. The reason I'm asking this question because when the IHCC submitted the first application, the reviewers commented that they didn't see in the application if outside of the IHCC, the investigator can access to the IHCC data. So since CanPath is one of the IHCC cohorts, so I asked a question, which is very good, and then other people can access the data as well.
Absolutely, yes. And so I think this is it speaks to future directions for IHCC as well, is that we'd like to be able to eventually move beyond just the data portal and talk more with our population cohorts about how we can make more visible their access policies, make more visible to some extent also the research projects that have been that are out there using those population cohorts as well.
That's great. Thank you so much, Peter.
Great, thank you, Rongling. I think the next question is about providing more information about air pollution and gene expression. Would you be able
sorry,
to...
I didn't have time. Oh, sorry, I didn't. Is this on the chat or I didn't?
It was in the chat from
I haven't
Dr.
opened up the chat yet.
Satyan.
Okay.
Yes.
What are the? Well, naturally, I can point you to the Nature Communications paper, which will do a much better job, of course, than what I could do in terms of describing this. But one of the things that we tried to do is in an unbiased way explore all of the variables that we could get our hands on through Canoe at that 6-digit postal code resolution, starting with 3-digit and then where we could 6-digit postal code resolution in Quebec, and ask what were the most significant variables explaining some of the gene expression variability data. And so while we did see certain groupings, like for example, green space was clearly an important variable associated with say gene expression by itself. as well as some of the specific genes that we were seeing. So we were actually seeing quite a lot of genes pop up associated with that have been previously associated with, say, COPD, which makes some sense. It still came down to things like PM2.5, NO2, and SO2. And that was able to help us separate, say, variability or explain variability across cities like Saguenay, Montreal, and Quebec. But we could also see, because we were looking at 3- and 6-digit postal codes, that we could see even within a city like Montreal, for example. So Montreal is an island, and we could see different things between, say, the North Shore and the South Shore, or Montreal Island versus the South Shore, in terms of how particulate matter, for example, varied and impacted, say, gene expression and as well health outcomes. And one of the ways that I wanted to highlight in my presentation there is that we're able to control for genetic ancestry. So we can quite clearly discriminate how much genotypic variability was explaining that variation versus, say, environmental exposures. And that has a lot to do with the fact that we were using a French-Canadian data asset to support that as well. So I don't know if that is answering the question, but again, I want to highlight that the canoe data, those environmental exposures are all now linked across the CanPath cohort, and you can get access to it through the CanPath portal as well.
Great, thank you. I don't know whether anybody else has a question before.
Hi, Philip, Scott. Can I ask you a quick question? So,
Scott, go ahead.
very exciting data on the pre-diagnostic methylome information. I'm just wondering what the implications are for cancer treatment of patients going forward. If now you can diagnose them before visible tumor through imaging or some other method is used.
Yeah, it's an important question with regard to, I think, a lot of what's happening in both the industry space and the clinical space as well. Sure, we can potentially predict with some confidence and relate that measure of confidence to a participant. You could buy a Grail kit, for example, and they'll give you some sort of risk assessment. But what do you do with that, right? And to some extent, this comes up in a lot of these conversations that we're having across in the UK and our colleagues in Oregon and Stanford around, you might be just increasing anxiety without there being an actual treatment available. It does certainly speak, though, of course, to potentially pushing a participant to more active screening. That's clearly an outcome. And also, speaks to, I think, this point that early treatment is the best way to address these cancers. You want to prevent these cancers from developing at their earliest stages. And we know the efficacy of even existing treatments are more efficacious in that space. And also, there's still a lot to be said for exploring or developing new treatments at these earliest stages, because traditionally we capture in biopsy, sequence the genomes and exomes of things when we see them, rather than at these even earlier stages. So, that's in the context of things like breast and prostate. For something like pancreatic cancer, right, where I'm continually hearing from clinicians that they need to be armed with something that's going to be able to get a patient in there before these things are stage 3 and 4. And so I think there's where there's got to be a lot more clinical application, because that's where the greatest urgencies are. When something's, because we don't even know when something is stage 3 and 4, what's developed in between, where these are multiple hits, whether stem cells have developed or not. There's just a whole lot of unknown in that space for some of these very rapidly evolving cancers as well.
Thank you.
Philip, can I just add to Scott's question, ask about both the methylation profile, but also the Intermountain risk score, and how potentially these could be used in screening. So, for example, in breast, if you are detecting before mammography, or even trying to see, at the time of the mammography, whether the methylation profile will triage women to those that need to have more frequent screening and at high risk, or high risk of a fatal disease, or whichever way, and then in a way using both scores within a breast screening program to address those questions. I don't know whether you have, thought about this or have any pilots or any data
Yeah, so I think this is a nice integration of multiple projects that we have here. Like, for example, Nick's project, we tried to see if there were other variables that we can incorporate into his modeling, apart from, say, just the DNA methylation that we captured from plasma that would help in the predictiveness. Rather remarkably, and this is just a bit of a teaser, things like CBC were not, or blood counts were not that helpful. One of the things that we can see nicely with Nick's data is the things like in other environmental exposures like alcohol usage. So we can see, a frequent alcohol user versus a non-frequent alcohol user in the blood profiles of those participants. And alcohol, particularly in Canada now, we just heard we had some things come from the Canadian Research Society that alcohol risk is a significant risk even beyond just 2 drinks a week for things like breast cancer and prostate cancer. With that said, in Alyssa's work, we were also able to say that CBCs were somewhat predictive for all cancers as well, complete blood counts. So in that healthy aging group, that and participants there were actually selected such that they didn't develop any sort of chronic disease or cancers. But when we followed them, we saw that people in our high-risk CBC group, some of them did go on to develop cancers as well. So I think there's some work here to do to integrate all of this information that we're capturing. Time will tell whether these things are complementary or are they just or if they're not doing that much better than looking at these things individually or alone. So.
And I know that you mentioned that you have obviously the biobank of pre-diagnostic samples. Do you have longitudinal sampling, or it's one per participant?
For some individuals, we've had, and that's been more budget restraining, of course, because in fact it was a very multi-million-dollar program to collect a single time point. But we're now going back out in the field. And in some cases, so in Quebec and British Columbia, they've done a significant amount of recontact, particularly in British Columbia. Here in Ontario, we're doing recontact for blood samples as well to get, and we're happy to hear suggestions on this. We're from our perspective, we want to target individuals that we think are more risk or high risk. Now, with that, with regard to development of disease, and that risk might be associated with environmental exposures. One thing I will highlight is that in the context of COVID, we've been able to go back and get frequent blood samples for many participants in CanPath as well. So the pandemic actually initiated, in some cases, 3 or 4 blood samplings from many participants. So I'd say right now we have about 30,000 of our participants where we've got a lot of longitudinal blood samples right now. We have a lot of stuff now in the bank, and so now we have to figure out how to put them in, turn it into data, actually. So
that's great, thank you. Cindy Lawley has a question. Cindy, go ahead.
Hey, Philip, great presentation. What a fun ride. My question is about doing this within the Canadian healthcare system. I'm in the US, and in the US we are very fragmented, right? I'm wondering if there were aspects that made it either advantageous to be in a more, I don't know how to describe it, but a different healthcare system than ours, advantages or disadvantages, either in building this population cohort or even what you see is needed to move some of the discoveries to the clinic.
Great question, Cindy. And so I think there are both advantages and disadvantages. Some of these lie in the fact that Canada is a confederation of provinces. So in general, the way healthcare works in Canada is we have 10 provinces with 10 healthcare systems. And 10 healthcare systems means you have 10 privacy officers and 10 different administrators of health data. So I don't know, I think that's potentially less fragmented than in the US system where you have different providers of health. But what that also has meant, though, that any contract that we've tried to do, for example, in building up a national activity, has meant that we've had to talk to numerous stakeholders to get support for this activity. With that said, either by design or perhaps by accident, in CanPath, we consented to participants to both national and provincial data. So that might have been fortuitous, because now, while we have consent to link to administrative records at the provincial level, we are all working now with some organizations that have been around for an awful long time, supported by Health Canada, to aggregate health information across Canada. And so we're very excited now about a new partnership with the Canadian Institute for Health Informatics, which has captured that national data. And now we're going to be able to link out to CanPath data in a much more streamlined way, so that for any research scientist, they're not going to have to talk to necessarily 10 different provinces. Now we're going to be able to just work with one entity. And that, I think that's going to be very exciting for Canadian research, or even international research, because that's been like the holy grail that I've been trying to work towards for the past 12 or 13 years as well. So we're really excited about that. Kim Skeed, who I mentioned, her research and is a national scientific coordinator for CanPath, has been a driving force behind helping generate that contract. But again, a lot of that sometimes is just also, Kim's been persistent and persevering for that, but also we're very excited with Kai, that Kai has listened to the Canadian research environment, who has been saying, we need this, right? We need to be linking out to national linkages. And so we just happen to be quite lucky in CanPath that our consents allow that at a national level as well.
That sounds promising. Thank you so much.
Thanks, Cindy.
I don't know whether anybody else had any questions, but
I do see one last question from Moeen Riaz in the chat. CBC
Yes.
for health and aging and not other biomarkers such as lipids. Part of the problem with lipids would have been great, but it wasn't something that was specifically interrogated, let's say, or used by the Intermountain Risk Score. I think I'll have to go back. I think blood sugar levels were for sure. I'm probably remiss in saying that lipids weren't. One of the things I will say is that CBC was an easy sort of first pass, and I knew I would have information that was easily harmonizable across Canada, in part because we had some of the same providers capture that information, exact labs, whereas there's going to be some significant work on the lipid front. With that said, that's certainly something we can explore as well. I just highlight that the RDWCV, red blood cell with the coefficient of variation, is an interesting feature of blood that seems to be highly predictive of blood health. And we're not the first to see that. In fact, we just can confirm something that was seen by our colleagues in Israel when they've been looking at similar types of data assets there as well. So I'm not really answering your question, but I do think that's an interesting way of thinking about what things we can incorporate into these tests. Sorry, Alex. Yeah.
No. Thank you so much. And I'm sure I'm speaking on behalf of the entire audience. That was absolutely excellent presentation, a lot of data and a lot of, I guess, possibility to do future work and follow up the cohort for a long time. So I'm sure that people will probably be approaching you for collaborations, but this has been a really absolutely amazing highlight of the. Oh, Latifa has a question. Oh, probably a comment for the next webinar.
No, actually just a comment for the next webinar. So thank you again, everyone, for your participation to this excellent presentation by Professor Awadallah. Please mark your calendar for the upcoming educational webinar series that is going to take place on April 26th at 9 a.m. EST. By Dr. Gelt Kelchersmans, and the title of the talk is going to be Leveraging Public Data to Understand Genetic Sensitivity to Air Pollution in Pediatric Asthma. So looking forward to your presentation, and thank you again. I think we are at the end of this session, Alex. So
thank you, everyone.
Thank you.
And yes, and the recording will be available for your colleagues to view if they've missed the talk. So thank you again, Philip. This was a great session.
Okay, yeah,
Thanks,
I'll be
everyone.
sharing the talk very soon. Thank you. Bye-bye.
Thank
Bye-bye.
you.
Bye, Philip.
Bye.
Bye.